For all of human history, the first few weeks after fertilization have been a total mystery. It's been this biological black box. But what if we could finally peek inside? Today, we're going to dive into a revolutionary technology that is allowing us to model the most complex, the most vulnerable moments of our own creation, right from the very, very beginning.
To really get why this is such a huge deal, you have to start with this one staggering number. Only about 1/3 of all human conceptions actually make it past the first few weeks. Just think about that for a second. Most journeys of life, they end almost before they even begin. That shows you just how fundamentally vulnerable we are, right at our origin.
So that brings up the huge question that has driven researchers for decades. Why? Why is this period so incredibly fragile? For so long, scientists have basically been working blind, trying to understand this time when the fundamental blueprint for a human being is being laid down, but it's all been happening completely hidden from view.
And look, this isn't just some abstract academic question. The fact that we can't study these first few weeks has very, very real consequences for human health. We're talking about everything from infertility to devastating congenital conditions. Opening this black box, well, it could help us solve some of medicine's most profound and difficult problems. Let's just put some numbers to this, because the stakes here are incredibly high. Infertility, that affects about 1 in 6 couples. And even with amazing tech like IVF, the success rates are still only around 30 to 35%. On top of that, so many serious birth defects trace their origins right back to this tiny window of time. It's also when an embryo is most susceptible to toxins, so our inability to study this period isn't theoretical at all. It has deeply personal consequences for millions of people.
So you might be wondering, how have we learned anything at all about this? Well, for decades, our only direct window into this whole process has been through human embryos that were donated from fertility clinics. And this has been an absolutely invaluable resource. But it comes with some major limitations. The supply is incredibly small, and it's also subject to really strict ethical and legal rules that limit what researchers can actually do.
And the biggest constraint by far has been something called the 14-day rule. It's been a cornerstone of research ethics for about 40 years. Basically, it sets a hard limit. You cannot culture a human embryo in a lab for more than 14 days. That point is marked by something called the primitive streak, which is the very first sign that the body plan is starting to form. Now, this rule gave us a vital ethical line in the sand, but it also created a scientific wall, completely blocking our view of everything that happens after day 14.
So how do you get past a wall like that? Well, this is where the story gets really incredible. Scientists have developed a whole new kind of model that lets us study this black box period on a scale that was totally unimaginable before. We're talking about going from a few precious little glimpses to literally thousands of reproducible experiments.
They're called Stem Cell-Based Embryo Models, or SCBEMs. Okay, let's break that down. These are not embryos made from an egg and a sperm. No way. They are three-dimensional structures built entirely from stem cells in a lab. The best way to think about it is like a biological flight simulator. A flight simulator isn't a real airplane, right? But it's so sophisticated it can teach us how a real one works under all sorts of conditions. It's the same idea here. These models are not real embryos, but they mimic the key steps so well that they can teach us how life's blueprint actually gets put together. So how do you even build one of these? Well, you start with what are called pluripotent stem cells. These are the master cells that have the potential to become any cell type in the body. Then you just add a few chemical signals, like a simple set of instructions. And here's the wild part. The cells do the rest. They just self-organize on their own, assembling into a structure that looks and acts a lot like an early embryo. It's pretty amazing.
Now let's be absolutely crystal clear on the difference here, because it is so important. A human embryo is made by fertilizing an egg with a sperm, and it has the potential to become a fetus. An SCBEM, on the other hand, is built from stem cells. It has absolutely no potential to become a fetus in its current form, and it's made purely for research. And because you can create tons of them from stem cell lines, they allow for the kind of big, systematic studies, like testing hundreds of drugs or screening for genetic issues, that would be completely impossible with donated embryos.
And that ability to do research at scale, well, it just blows the doors open on what's possible. This technology could seriously unlock breakthroughs that could transform medicine, not just by helping us understand our own origins, but by helping us build a much healthier future.
Take a look at this map here. It lays out the whole journey of what this could mean. If you see on the far left, it all starts with fundamental research into the very earliest stages, like how an embryo implants in the uterus, which has huge implications for IVF. Then, as we move along, the models get more and more complex, letting us study how organs begin to form.
And that opens the door to all the things you see at the top: modeling specific diseases, testing if drugs are safe, and even, hopefully, regenerative medicine down the line.
So, why does this matter to you? Well, it means we could finally figure out why so many IVF cycles fail and maybe come up with better ways to improve those success rates. It means we could understand the signals that go wrong during implantation and lead to miscarriages. We could literally create a model of a congenital heart defect to see exactly how it happens and maybe learn how to prevent it. We might even one day be able to grow new tissues and organs. And so importantly, we can test new drugs on these models to see if they're safe for pregnancy, something that's incredibly difficult to do right now.
Okay, but, and this is a big but, a technology this powerful doesn't just solve old problems, it creates brand new questions. As we learn to model the very beginning of life in a dish, we have to start navigating a whole new landscape of really complex ethical issues.
You know, the central ethical argument for doing this research really boils down to intent. These models are not being created for reproduction. Their entire justification, their whole purpose, is to advance scientific and medical knowledge in ways that were impossible before. In some ways, they're actually an ethical alternative to using real human embryos or even animals for some types of research.
And that leads us right to the heart of the debate. As these models get better and better, more and more accurate in how they mimic a natural embryo, we have to ask ourselves, how close is too close? At what point does a model start to raise profound ethical concerns just because it's so similar to the real thing? This is the boundary that everyone, scientists, ethicists, and the public, has to figure out together.
So let's break down the main concerns here, because there are really three big ones. First, there's developmental potential. Could one of these models ever develop into a viable fetus? The answer right now is a clear no. They're missing key parts and have lots of abnormalities. But it's a question for the future. Second is what's called symbolic value. Even if a model can't become a person, what does it mean when we create something in a lab that develops a rudimentary beating heart? That can create a sense of unease. And finally, and maybe the most profound question, is the potential for sentience. If a model were ever to develop a complex, integrated nervous system, could it feel? This is a distant concern for sure, but it's a serious one that helps guide the limits of the research.
So, with all these questions on the table, how do we move forward responsibly? Well, the good news is the scientific community isn't just ignoring these issues. They are actively working to build a framework to make sure this powerful science moves forward ethically and with public trust.
This diagram here really lays out the proposed roadmap. If you look at the bottom layer, it all starts with standardizing the models themselves, how they're made and what they can do. Then, that standardization, shown in the middle layer, allows for clear and effective regulatory oversight. And it's only with that strong oversight in place that we can responsibly move to the top layer, turning this amazing research into real medical benefits for people.
So what does this actually look like in practice? Well, first, standardization, making sure everyone's on the same page with materials and methods so that results are reliable. Second, strong oversight by ethics committees that really know this specific area. Third, some very clear, hardline prohibitions. There's a global consensus that transferring one of these models into a human or animal uterus should be completely banned. And finally, and maybe most importantly, an ongoing public dialogue. Scientists need to be open about what they're doing and why, and listen to the public's hopes and concerns to build trust and decide on these boundaries together.
And that brings us to the big final thought here. For the first time in history, we have the tools to decode the very first chapter of the human story. But this incredible new knowledge forces us to confront a question that goes way beyond any single experiment. As we learn how to model the beginning of life, we're also being asked to decide how we define the boundaries, not just of a biological model, but maybe of science itself.